Hello and welcome to this lesson for higher biology, which is the second part of key area 2 of unit 1. So we're looking at DNA replication, and in this case we're going to be looking at PCR or polymerase chain reaction. So prior to now, we were looking at DNA replication within the cell and the reasons why DNA needs to be able to replicate within the cell and the processes that are going to occur, including the enzymes and the different elements that are involved in there. What we're going to be looking at today is a process called PCR, which is essentially replicating DNA in a tube in a lab rather than doing it in a cell. And there's quite a few different reasons why we might want to do that. We'll move on next week to be looking at gene expression, so how genes are actually producing proteins within a cell. But for today, we are looking for you to understand what the process of PCR is and how we use it. You need to be able to explain the temperature cycling process in PCR. You need to be explain why primers are important in PCR, what is specific about the polymerase enzyme used in PCR, and be able to give some examples of how PCR is used. So PCR itself is something called the polymerase chain reaction, and it's basically a technique for amplifying DNA, so growing it and making more copies of it in vitro or outside the body. So it's a process by which we're taking a DNA template and we're adding useful things to that DNA template in order to replicate the DNA. Now, it's particularly important to be aware that we are not necessarily, or not usually, when we carry out a PCR, looking to copy the entire molecule of DNA that's inside the cell. Normally what we're trying to do is we're just trying to copy one particular section. It might be a specific gene that we're looking at, that we're interested in researching. It might just be a particular area which tends to be quite different from person to person if we're looking to try and do some identification or paternity testing. Or it might be looking for something really quite specific, and we'll look at that when we come on to some examples. So in order to carry out a PCR, you need a supply of primers. Now, because we are looking usually in a PCR at amplifying or making copies of a very specific section of the DNA molecule, the primers are normally designed in advance, and these are going to be quite specific to the area of DNA that we want to copy. We also need to provide a supply of the nucleotides. So we need a nucleotide with each of the 4 bases on it. So we need several A, several T, several C, and several G, so on. We need something called a pH buffer, and that's just to make sure that the reaction doesn't get too acidic or too alkaline. It maintains its pH and doesn't cause the reaction to stop. We also need a DNA polymerase molecule, remember, and that's the one that adds our free nucleotides to the 3' end of our new DNA strand. So we need it to go in there, and we need our original DNA template in order for it, or sections of it, to be copied. So as was said, in PCR, it's usually a very specific section of DNA that's to be copied. So the primers are designed to be, by the scientists, to be complementary to that specific target sequence. And as a result, if we use those primers, it's only that area of interest that's actually copied. So if we just wanted this little section of our DNA here copied, say this is the gene that we are interested in, what we would do is we would design a primer, one going in one direction, one going in the other direction, so we can copy both strands. So as you can see, this one here is complementary to this strand down here, and this one here is complementary to this strand up here. So that means that when these DNA templates are split apart, this one will come along and bind on to this bottom strand, and then the DNA polymerase is able to add the new nucleotides on to that section. Similarly, if we've got our reverse primer here, it's been attached to this top strand, when those ones are separated, we'll have our DNA polymerase will be able to come along and add more nucleotides along here. And those primers, because they're specific to that particular area, just means that we only need to copy that bit of DNA. We're not going to copy the whole molecule. Now the process of PCR is actually quite simple. We begin with heating the DNA to separate the strands. So obviously in the cell we've got DNA helicase responsible for carrying out that particular role. We're not going to use that enzyme, we're just going to use temperature. So we've got all of those items in that little tube. We've popped it into a machine which is called a thermocycler. And what we're going to do is we're going to change the temperature several times and go through a cycle of different temperature changes in order for different things to happen. So step 1 is heating it to between 92 and 98 degrees, and that's going to heat it up and separate those strands. Now we need to cool it down a little bit to let the primers, which are correct for our sequence, are complementary to our sequence, to be able to bind. You might also see this referred to as the primers annealing to the template strand. So as a consequence, our second temperature is between 50 and 65 degrees, so cool enough to allow those primers to bind on to the complementary DNA. In step 3, we have our free supply of nucleotides, and we have a specific heat-tolerant DNA polymerase, and that's used to replicate the DNA by adding the free nucleotides onto the primers, just in the same way as DNA polymerase does to our primers in cellular DNA replication. So we have our DNA polymerase here coming along to add our nucleotide onto the primer, and we've got another molecule coming along here. This is all happening between 70 and 80 degrees. Now, remembering normally an enzyme would denature above sort of 40-45 degrees or so, and that's why these specific DNA polymerase enzymes are heat tolerant. They're able to withstand those particular high temperatures and operate under those conditions. So step 4 is essentially going back to step 1. We're going to repeat it over and over and over again. We've got double the DNA that we had, and we're going to go back and split those DNA molecules up and let primers anneal to them and duplicate those again and again and again, and we're going to continue to multiply our DNA strands. Once we have completed the PCR, we normally take the DNA that's been completed and it gets put through something called a gel electrophoresis. And essentially what this means is that different sized pieces of DNA are able to move at different speeds through the gel. So if we have these hundreds and thousands of sections of DNA which have been copied by our PCR process, then that tube is going to have lots and lots and lots of DNA in it. So it's going to have a large amount of DNA present in that tube. If the testing tube, the initial DNA template that was put in, did not have the area of DNA that was complementary to the primers that we were able to copy, then it would not be able to make all of these copies of that section of DNA, so we wouldn't have that much DNA. So if that specific area of DNA has been able to be copied by the PCR, if it was present and the primers have bound to it and we've copied it, we've got lots and lots of copies of it, and as a result we're able to identify the pieces of DNA by size. Now, using PCR, we might use that for research in order to look into specific genes and the sequences of specific genes and how they may influence disease or drug responses. We might use it to look at population studies, so taking a population and looking into some inheritance and looking into some background information as to where these populations originally arose. We might use it for anthropology, so detect it using PCR to detect specific areas of DNA which are really relevant or linked to individuals alive today, so we can maybe identify individual remains or identify family groups of individual remains. We may also use it for disease research, so looking at potential genetic links into different diseases and looking at it in terms of how individual mutations in DNA may be causing or not causing different diseases. Now, there is a current use of DNA which is actually involved in COVID-19 testing. So I'm going to talk about that a little bit on the next slide. If that's not something that you want to hear about right now, that's absolutely fair enough. So you can really just stop the video here. That's the last little bit you needed to know about maybe skip forward a minute or so until we get to the final slide again, as I'm reviewing the success criteria if you would like to. I'm not going to talk about it in a huge amount of detail, I just wanted to give you a little bit of advanced warning. So at the moment we are actually currently using PCR a lot in order to test individuals to see if they have COVID-19, and I just thought this was worth mentioning because it just explains a little bit about how PCR actually works. So essentially what we would do is you're going to take your specimen swab from your individual. That is going to contain a whole bunch of cells. So it's going to contain their cells from the back of their throat, the back of their nose, and if that individual has been exposed to the coronavirus and we can detect that, it will also contain some viral genetic material in here as well. And it's important to realise that in this particular case, the individual viral material tends to be RNA, not DNA, but don't worry about that for the moment. The principle's the same. So when we've extracted those cells, so we've got human cells and we've got viral cells in here as well. And what we can then do is we can then take those cells out, we break them apart, and we take the DNA or RNA, any genetic material, we're going to take that out. What we then do is we've designed primers which are specific to the genetic material of the virus. So those primers will only bind to genetic material which is viral genetic material, and they won't bind to any human genetic material. So as a result, the only bits of DNA which are replicated or amplified are the bits that have viral DNA in them. So again, if you consider that gel electrophoresis, if we've got hundreds of copies of viral DNA, maybe they are this size here, maybe they're 800 base pairs. So when they have been copied and that is run out on our gel electrophoresis, you would be able to see that these two samples would be testing positive for having that length of DNA, which means that's the viral DNA length. So they'd be testing positive for having those bits, and this individual would be testing negative. So when we look at those results, that's essentially what they're doing, is they're looking to see, do they have that band or that size of DNA or genetic material that indicates there is viral material there, in which case that individual does indeed or has indeed been exposed to the virus. If they do not have that particular section of DNA, which would be viral DNA, then they haven't been exposed to it, or they don't have enough viral particles present to be picked up. So I just thought that was worth mentioning it, as it's particularly interesting at the moment. So from that, you should be able to understand that we've got different temperatures involved in PCR in order to break apart our DNA molecules, allow the primers to bind to the original DNA template, and finally to come back and let the DNA polymerase replicate those specific DNA sections. And that process is repeated over and over and over again, usually between 30 and 40 times, in order to amplify or replicate that original DNA section. But the primers are of ultimate importance in PCR, as they are specific to the bit of DNA that we are interested in, or the bit of genetic material that we're interested in, and they're complementary to that area, so only that section of DNA will be amplified or replicated. That the polymerase enzyme used in PCR has to be heat tolerant in order that it does not denature when we get into those high temperatures, and that PCR can be used, well, currently in viral testing, or it could be used for population studies or disease research, or looking at anthropology and inheritance information.